We are going to be talking about the cell division, which is the daughter cells when they do divide, you having the same genetical material and from the parent cell, and they do form a complete set of chromosomes. Now the first step is going to be called the interphase, which is when the cell starts to divide. This is the longest phase though. During this time, the chromosomes do replicate. So the DNA strand unzips into two strands, and then it just has two floating straps, let's say. So in this case, the two pair of centrioles, which are going to be those dots as they do show in the image, they are just left outside of the nucleus. And the centriole is a cylinder structure type of within the cell that is just plays part in the cell reproduction. The next step is going to be the prophase, in which the chromosomes do start to condense. They do take the form that that is showing above or in the image, which has like four arms that are connected at a point, not in the middle. Now each chromosome at this time has two identical copies, and each of one of those copies are called a chromatid, which is gonna be this image in the that is showing the circle in the middle. It's what connects them, which is the centromere, and the two strands on top are gonna be called the sister chromatids. At this point, a spindle begins to form from the centrioles, and this spindle is made of fibers. Now the centrioles do begin to separate, and the membrane of the nucleus or the nuclear envelope fragments, and they disperse. Now the spindle fibers are protein structures that pull apart the genetic material in a cell when the cell does divide. Now we go into the M phase or the metaphase, in which the centrioles are now at the opposite ends of the cell, and the spindle fibers from both the centrioles do attach to each one of the chromosomes. Now the chromosomes start to line up inside the metaphase place in a little imaginary line, and it divides the cell into two. Now the spindle fibers, they do begin to tug each chromosome towards the opposite end of the cell. Next is going to be the anaphase, in which just the spindle fibers, they just pull the chromatids towards the opposite end of the cell. Now the telophase, the next step is just when the chromatids that are now considered chromosomes, they do arrive to the opposite end of the cell, and the new nuclear membranes do form. In the next step is going to be cytokinesis. In this step, the rest of the cell divides, and the division of the cell's cytoplasm is complete. So the entire process has been complete with a result of two identical cells.
Why cells divide? Our organism must produce genetically identical daughter cells. Single-celled organisms do this to reproduce. Each of the produced cells is a separate organism. Multicellular organisms divide cells for three reasons: growth, repair, and replacement. Multicellular organisms can grow in two ways: by increasing the size of their cells or increasing the number of cells. This last option is achieved through mitosis. Mitosis is a crucial part of the entire cell cycle because it's a point at which the cell passes its genetic information into its daughter cells. Division also makes sure that the new cells are available as replacement when older cells are within the organism die. When cells are damaged, they need to be repaired. They are replaced with identical cells capable of doing the exact same job. All cells need to be replaced at some point in their lifetime. Red blood cells last for about 3 months, and skin cells even less. Identical cells continue the job of the cells that they replace.